Next topic is important biocontrol project undertaken in India by employing predators and parasites. Introduction: the successful management of pest by means of another living organism, parasitoids, predators, and pathogen that is encouraged and disseminated man is called biological control. In such program, the natural enemies are introduced, encouraged, multiplied by artificial means, and disseminated by man with his own effort instead of leaving it to nature. So, biological pest control project in India, the first international effort was made in 1873 with the export of predaceous mite from the United States to France to control the grape aphid Phylloxera. The first international effort was made in 1873 with the export of predaceous mite from the United States to France to control the grape aphid Phylloxera. In 1887, worldwide appreciated trials was made by exploring the ladybird beetle from Australia and its establishment into California for the control of cottony cushion scale. By receiving encouragement from the above achievement, exotic predators and parasites of the pest species were collected from the native home and introduced into the affected areas for keeping the pest in check by means of biological control. In India, the first Indian Station Commonwealth Institute of Biological Control (CIVE-PC) since has done a pioneer work in controlling some serious pests by introducing their parasites and predators from foreign countries. Some of the initial successful biological trials are cited. Control of Opsina arenosella. Opsina is a coconut pest. Its larvae, called black-headed coconut caterpillar, is seriously harmful to coconut palms and its control is very difficult. Biological control method has been proved to be effective in this case. The parasitoids Gonios nefandidis and Trichospilus pupivora, Bracken bravicornis, and Clasmus nefandidis are the natural enemies employed in this method. These natural parasitoids are multiplied in large numbers in parasite breeding stations and then released to pest-affected plantations. Goniosis attacks a grown-up caterpillars of the pest and it is a restricted parasite affected only in Opsina arenosella. In the parasite breeding station, the adults of Goniosis are kept in glass tubes of 15 into 2.5 cm size. Small bits of waxed paper dipped in sugar solution are introduced into each tube for the parasitoids to feed upon, and every day new paper bits are introduced. Small bits of waxed paper dipped in sugar solution are introduced into each tube for the parasitoids to feed upon, and every day new paper bits are introduced. Mating takes place inside the tube. The mated females are separated and placed one on each tube. Also, these tubes grow up caterpillars of the pest are introduced. The female parasitoid lays eggs on the caterpillar and the eggs are glued to the sides of the caterpillar. After about 36 hours, grubs hatch out and they feed on the body fluid of the host caterpillar. This continues for about 5 days, which is the time the caterpillar dies. These parasitoids are taken to the top of the coconut plants and released at the rate of 10 to 15 parasitoids per tree. Control of Escherichia berkshire. It's a very common polyphagous pest affecting fruit trees in Western countries. In 1928, this pest somehow got introduced to Tamil Nadu. It spread all over Nilgiri and became serious pest on the vital Acacia dacrynus. Its natural enemy, the beetle Rhodolia, was brought from California in 1929 and multiplied in laboratory. These beetles were released into the plantation and within about a year the pest was successfully controlled. In 1942, then the same pest again occurred in Kodaikanal Hills. Kodaikanal Hills. The same beetle was used to check the pest outbreak. Control of Chilo infusculatellus. Trichogamma coleonis, which attacks the egg of Quail, is used in the biological control of the latter. This parasitoid is released into the field through the parasitized eggs of another host species, Corcyra cephalonica. First, the moths of Coursera are bred in the laboratory on sorghum grains. Both male and female moths are grown under the same inverted funnel. A piece of white cardboard is placed inside the funnel. The female lays eggs on the cardboard. Next day, the eggs on the cardboard are sprinkled over a wet paper and allowed to dry. These egg cards are kept in a glass tube into which the parasitoids are introduced. The eggs get parasites and become black in color in 2 days. These egg cards are hung in the sugarcane fields. The parasitoids came out of the eggs of Cephalonica and attacked the eggs of the pest Chidioinfescatellus. Sugarcane pyrilla. Tetrastichus pyrillae and nymphal predator Epiphyrops melanoleuca. Apple woolly aphid and San Jose scale.